So in this lecture, we're going to be kind of rounding out affiliative behavior by talking about the ways in which hormones affect sexual behavior. We're going to be focusing on male and female sexual behavior, but starting out with female sexual behavior. If we were to kind of utilize human beings as our model to understand sexual encounters, I think we would be at a complete loss, largely because there are many factors that go into mate choice. It's not just attraction. There are many psychological as well as physiological factors that influence females' mate choice of males, and it's a complicated behavior. It's who you encounter, who you're comfortable around. It's not just simply, is this an attractive mate? And so it's really difficult to study the mechanism that controls sexual mate choice in human beings. So instead, we end up using animal models, such as the rat, to better understand how this process works. The rat female estrous cycle is what governs female sexual behavior. And so the estrous cycle is analogous to the human menstrual cycle. But unlike the human menstrual cycle that's on average 28 days, the rat estrous cycle lasts only about 4 days. And the hormonal changes that occur during the estrous cycle are characterized by 4 stages: proestrus, estrus, metestrus, and diestrus. Here is the pattern of hormonal changes that occur during the 4 different stages of the estrous cycle shown here on the bottom. LH and FSH are pituitary hormones, and progesterone and estrogen are ovarian hormones. But they're exactly the same hormones that are released during the human menstrual cycle. So the human menstrual cycle lasts roughly 28 days on average. But you'll notice that LH and FSH go up as well, along with estrogen, and then you have a later peak of progesterone levels. You'll notice that the estrogen levels go back up again in human beings that you don't really see in the rodents, but really, other than that, they're pretty much identical in their pattern of release. So we're going to be using the rat female estrous cycle as a way to better understand the ways in which hormones regulate sexual behavior as well as potential changes in the brain. So what's interesting is that the changes that occur in these hormones, while they serve a reproductive function, which is the release of the egg or the follicle, they actually produce changes within the brain. So researchers have found that as you change these hormone levels in the brain, hormone levels in the body, they produce changes in the dendritic spine or in synaptic density in an area of the hippocampus when estrogen levels are high. So during proestrus, right, just shortly before the follicle release, estrogen levels are really, really high. This results in increases in dendritic spine and synaptic density. So dendritic spines are those protrusions that exist on the outside of dendrites that increase the surface area for synaptic contact with other neurons. So just to illustrate to you what these dendritic spines look like, the arrows are pointing to the dendritic spines here. So this is a dendrite that you're looking at. And here is the dendritic spine of a female during proestrus versus here's the dendritic spine of a female during metestrus or diestrus. You'll notice that there are many, many more spines during proestrus than during metestrus or diestrus, suggesting that estrogen seems to regulate synaptic density. And these are changes that are not permanent. They change over the course of the menstrual cycle, and researchers have been able to administer estrogen to female rats and produce exactly the same changes in synaptic density. So we know that it's estrogen that's actually producing this effect that we're seeing with the dendritic spines. If you remember, the hippocampus is important for learning and memory and spatial navigation. And when they've tested female rats over the course of the cycle, they found that it corresponds very nicely with cognitive strategies that rats use on learning tasks over the course of the estrous cycle. In addition to the reproductive functions for these hormones in the rat, there are clear changes or effects of them within the brain as well. So sexual behavior in females is regulated by the release of the egg. So the female becomes interested in copulation or sex, essentially after the release of the egg, which occurs between proestrus and estrus, or the transition between proestrus and estrus. In the male, copulation consists of the male mounting, intromitting, which is essentially inserting his penis into her vagina, as well as ultimately ejaculating. These are elements of the male sexual behavior. Females demonstrate two types of behavior. One is reflexive, that's lordosis behavior, shown here. So lordosis is this arching of the back that the female takes on, and essentially it is to allow the male to intromit into her vagina. So she's exposing her vagina to him. This particular behavior is reflexive, so she doesn't really have control over it because she demonstrates this behavior when the male mounts her or grasps her flanks. What does seem to be under her control is what's known as proceptive behavior. The female can solicit male's sexual interest through proceptive behavior to let him know that she's interested, and the proceptive behavior is also regulated by hormones, but includes behaviors such as hopping and darting and ear wiggling. So these may seem like silly behaviors to you, but essentially it's analogous to what humans do with flirting. They let the male know that she's interested and that she's in the right stage of her cycle. What's also interesting is that females regulate the pace of copulation. Males and females mate multiple times over the course of the night, and males would actually prefer to have copulation occur at a much faster pace so that they can increase the number of partners they can have in one night. So let's talk about the different ways in which the brain regulates these two types of behaviors, sexual behavior in males and sexual behavior in females. So if a female is in the right phase of the estrous cycle and demonstrates proceptive behavior, the male will mount her, which is what you're seeing here. So this is the male, and this is the female. She's arching her back, and the male is mounting her and likely intromitting her. So he will engage in bouts of mounting many, many times before he actually ejaculates, and after about 5 minutes of rest, he will return to this copulatory behavior. If the female is still in the cage with him. He will ejaculate 5 or more times in his encounter with the same female. What researchers have found is there's a structure called the vomeronasal organ that detects pheromonal signals, and the vomeronasal organ is part of the olfactory bulb. Males and females both have it, but it passes this information through the olfactory bulb to the medial amygdala. From here, the information goes to the preoptic area, shown here and here, which is specifically involved in proceptive or flirting behavior in females, and in males it regulates copulatory behavior. From here, the pathways kind of diverge between males and females. The VMH seems to play a role in lordosis behavior because when this area is lesioned, the female no longer demonstrates that arching of the back that seems to be reflexive. Additionally, the reward system, which includes the nucleus accumbens, shown here in males and females, it's part of the reward system and is also involved in the behavior. And we know that sexual encounters are rewarding to females because she will actually walk across an electrified grid to access a male mate. In males, we know that the MPOA, or the MPN, regulates copulatory behavior because when we destroy it, it disrupts copulatory behavior. Pheromones are also playing a role in activating the system in males, and the rewarding system also seems to be implicated in male sexual behavior, as males will work to obtain a mate. When we compare these regions in males and females, so this is the male brain and this is the female brain if you didn't already notice, we can see that there's a lot of overlap, but there are also brain regions that are unique to copulatory behavior between the sexes. Following multiple ejaculations, the male seems to be exhausted and stops pursuing the female. However, if a new female is placed in the cage with him, it seems like he regains his energy to pursue this new female. This is called the Coolidge effect, named after President Calvin Coolidge that you see right here. Apparently, the president and his wife were touring a farm in 1924, and for some reason they were assigned to two different tours, with his occurring after hers. When Mrs. Coolidge was on her tour, the level of sexual activity demonstrated by a rooster caught her eye, and she asked the tour guide how often the rooster exhibited such responses. Many times, the guide said. So Mrs. Coolidge said, "Tell that to President Coolidge." When the tour guide mentioned this to the president during his later tour, President Coolidge asked whether this activity was with the same hen every day, and the tour guide replied, "Oh no, Mr. President, a different hen every time." So in his response, the president recommended that the tour guide share that information with Mrs. Coolidge. So this effect is likely adaptive for non-monogamous species to enable the males to impregnate many, many more females in one given night. Incredibly, the Coolidge effect was demonstrated by a guinea pig in 2014. So Randy was the guinea pig who escaped from his enclosure for one night and ended up mating with 25 females in one night and ultimately siring 180 offspring. So there's clearly evidence for this Coolidge effect that's been named after a US president. So behaviors are different between the sexes, so this implies that there must be differences within the brain between the sexes, not just in terms of the structures that regulate sexual behavior. So the brain regulates behavior, and perhaps there are sex differences in the brain. Most predict them to be in the hypothalamus because it regulates reproductive behaviors, and this is certainly the case. The MPOA, the brain area that seems to regulate proceptive behaviors in females and mounting behaviors in males, is larger in males than in females. Another area, the AVPV, which seems to be involved in the secretion of those hormones from the pituitary, are larger in females than in the males. But sex differences don't simply exist in reproductive controlling areas like the hypothalamus. They also seem to be present in non-hypothalamic regions, such as the hippocampus. In rats, males have a much larger hippocampus compared to females, but ironically or strangely, males have a smaller hippocampus than females in human beings. Similarly, the rate of serotonin synthesis appears to be different between males and females. What you're looking at here is the rate of serotonin synthesis. Anywhere you see red represents increased production of serotonin, and you can see that serotonin levels seem to be higher in males. Serotonin synthesis appears to be higher in males compared to females. This also seems to coincide with the sensitivity of glucocorticoids. So glucocorticoids are the stress hormones that are released when we have our flight or fight response, and females are more sensitive to glucocorticoids than males. Both glucocorticoids and serotonin have been implicated in depression, and this may be one explanation why there's a sex difference in the incidence of this particular disease affecting females more than males. So we're going to switch gears a little bit and talk about the biological evidence for sexual orientation and gender identity. The first of this is the genetic basis for sexual orientation. So typically when you want to demonstrate that there is genetic contribution to something, you look at twins. Identical twins or monozygotic twins share 100% of their genes because they have developed from a single egg that split after the egg was fertilized, versus fraternal or dizygotic twins have the same relationship to each other as siblings, for example, but they share the same prenatal environment. So by comparing the concordance rates, that is the amount, the rate at which a particular characteristic exists in both twins, we can determine which characteristics are contributed by genes versus environment. So in 1991, Bailey and Pillard studied homosexuality in men to determine the concordance rates for sexual orientation. In this particular study, they looked at just homosexuality, not bisexuality or any other orientation on the spectrum. So as I mentioned before, when you have a twin that's concordant, it means both individuals in the twin pair have identified themselves as homosexual, versus if they are discordant, only one twin identified himself as homosexual and the other one not. So here's the evidence for it. Identical twins had a concordance rate that was 52%. That means 52% of identical twins had pairs where both pairs or both individuals identified as homosexual, versus the fraternal twins only had 22%. Does this represent the genetic basis for homosexuality? If you said yes, that would be correct, because we have a greater concordance rate for the twins that share 100% of their genes compared to the twins that only share 50% of their genes. Bailey and colleagues repeated this study again, this time in homosexual women, and they found similar results, 48% concordance rates for identical twins versus 16% for fraternal twins. So this really supports the genetic contribution to sexual orientation, at least with respect to homosexuality. But is it all genetic? If it was, we would expect the identical twin concordance rate to be 100% because they share 100% of the genes. But clearly, because it's only 52 and 48%, it suggests that there are other elements that are at play to determining sexual orientation outside of genetics. So there are several structures in the human brain that seem to be related to a person's sexual orientation, and we've talked about the fact that there are sex differences in the human brain where males and females differ in the volume or size of a given brain area, and so it was kind of not surprising that researchers were curious to see if there were any differences in the brains of individuals that differed in their sexual orientation. And so below are a bunch of structures that differ in their size between heterosexual men and homosexual men. So we're comparing men according to their sexual orientation. So you should know the SCN, right? It seems to be important for circadian rhythms. In 1990, the SCN was found to be larger in the homosexual male compared to the heterosexual male. Again, you should know that these are in individuals who are deceased. They were, you have to extract the brain and measure the size of the SCN, so these are not in living human beings. And a structure in the hypothalamus called the INA3 is larger in the heterosexual male compared to the heterosexual female and the homosexual male. In fact, the size of this structure was identical between heterosexual females and homosexual males. And lastly, the anterior commissure was larger in homosexual males compared to both heterosexual females and heterosexual males. So this suggests that there might be some structural differences between people of different sexual orientations, but as I mentioned earlier, these individuals were deceased. It's really difficult to conclude that these differences in brain regions contributed to the sexual orientation of the individual, because it could easily be the lifestyle that they lived contributing to these differences in brain areas. So just to illustrate to you where the INA3 is located, so this is the human brain, and we're taking a coronal, we're cutting it in the coronal or frontal plane, and this is the ventricle here, the third ventricle. The optic chiasm is right here, and on either side is the hypothalamus, and here is the INA3. There are 4 total INAs, interstitial nucleus of the anterior hypothalamus, but the one that seems to be different between males and females, but also across sexual orientation, is INA3, and that's shown here. So it's a super tiny structure. And this is the study that was actually done to illustrate to you the differences between males and females. And so here's the male section, here's the female section, here's INA3 in a male, and it's almost absent in the female. And the same thing was found in homosexual men. They had next to no cells in the INA3. The volume of the INA3 was much smaller in homosexual men compared to heterosexual men. And that kind of is represented here in the sex difference, where heterosexual males had a much larger INA3 compared to heterosexual females. And remember, the heterosexual females had a similar INA volume compared to homosexual males. All right, another difference that seems to be predictive of sexual orientation, or related to sexual orientation, I shouldn't say predictive because we don't know for sure, is differences in finger length. So I want you to follow me here because it's a little bit bizarre, the fact that we would be using finger length to correlate to sexual orientation. Okay, so in the right hand, there's a sex difference in the ratio of the pointer finger, which is the second digit, and the ring finger, which is the fourth digit. Women have a greater 2d/4d ratio compared to men. What that means is so here's a female, the ratio of these 2 digits is closer to 1 than it is for males and females. Essentially, it comes down to the pointer finger in the male is a little bit smaller than the ring finger, and on females, the pointer and ring finger seem to be more similar. To calculate the 2d/4d ratio, you would take an arbitrary number, and I've made these numbers up, but essentially you would take the second digit measurement and the fourth digit measurement, divide it, and get a value for women, and you would do the same thing for men, and you typically find the ratio of females to be closer to 1 versus males, which is less than 1. So women have a greater 2d/4d ratio compared to men. What's important to note about this sex difference is that it's a hormone-dependent sex characteristic. It is a sex difference that develops in children as young as 2 years of age, and it's entirely dependent on testosterone exposure in males in the fetal stage and not later in life. The CAH females that we talked about in the previous lecture have a 2d/4d ratio that's similar to males. And if you remember, CAH females are exposed to higher than normal levels of androgens, and their 2d/4d ratio has become more like males. So this illustrates the fact that this is a hormone or testosterone dependent sex characteristic, and it's hormone exposure during development, because children as young as 2 years of age show this sex difference, right? If it was puberty, we would expect this sex difference to not be present in kids as young as 2 years of age and only in adults, but the fact that we see it in kids as young as 2 years of age suggests that this sex difference is produced by testosterone exposure during development. So in another study, they used this logic of the sex difference between males and females in finger length, and having it be dependent on hormone exposure during development as a reason to look at finger length ratios between homosexual women and heterosexual women. So the reason why they didn't look at homosexual males is because males are typically exposed to testosterone during development. So it's really not feasible to look to see what hormone levels are like in males, because it would be more or less from what they already have. Females, on the other hand, we know don't have any hormone exposure, so if we see any differences as a result of hormone exposure, they're going to be more sensitive to those effects. And so in this particular study, they looked at the 2d/4d ratio of homosexual women compared to heterosexual women, and what they found was that homosexual women had a lower 2d/4d ratio compared to heterosexual women. The 2d/4d ratio in homosexual women were similar to what they found in males, which was smaller than heterosexual women. So what can we conclude from these findings? It's a little bit difficult to say that hormone exposure contributes or causes differences in sexual orientation, but certainly it seems to be that there's evidence that hormone exposure in utero may contribute to sexual orientation, again, if we go through the logic of the finger length differences, but it's not absolute. There seems to be some support to sexual orientation being determined by biological factors as opposed to environmental. So regardless of sexual orientation, researchers more recently asked the question, do the brains of transsexuals who transition from their birth sex to the opposite sex differ in some way? And one study found a difference in the volume of a structure that's part of the limbic system, the bed nucleus of the stria terminalis. You probably saw it in the lecture that we talked about when we were discussing the limbic system last week. So what they found was the following. So they compared heterosexual males, homosexual males, heterosexual females, and male to female transsexuals. Okay, so you'll notice that homosexual males have a larger BST compared to heterosexual males, and that there's a sex difference where heterosexual females have a smaller BST compared to either heterosexual or homosexual males. Again, these are individuals who identify as males. But in those individuals who transitioned from male to female, right, so they identified as female, their BNST or their BST volume was similar to heterosexual females and smaller than individuals who identified as males. So these studies suggest that there are differences in the size of structures between people who differ in their gender identity, but whether these structures are the reason for the differences in gender identity or resulted from the lifestyle that they live, it's really, really difficult to tell. But clearly there seems to be differences between people of varying gender identities. Another evidence that demonstrates differences in brain structures of people with differing gender identities was the study that looked at diffuse tensor imaging. So diffuse tensor imaging is a pretty recent technique that utilizes the movement of water to identify the axons or connections between brain structures. So in this particular study, they were looking to see if there were any differences between the connections of structures in individuals who differed in their gender identity. And so the structure that they were looking at was called the superior longitudinal fasciculus. It connects the prefrontal cortex with structures in the temporal and occipital lobes. And this is what they were looking to demonstrate was different between people of differing gender identities. So there's a lot of bars here, so I don't want you to be overwhelmed, but I want to orient you to what the bars are. So the female to male is in green. The female and the male are shown here in differing colors, blue and I guess that's navy blue. But the transition, the transsexual, is shown here in green, and the structures that you should focus on is here. So the female to male transition had a superior longitudinal fasciculus that was identical to males and larger than that of females. So this kind of supports the idea that there are differences between connections of brains in individuals who differ in their gender identity. They found a slight difference in the forceps minor as well, where the female to male was intermediate to the male and the female. So again, these differences should be taken with a grain of salt, but clearly there might be some biological contributions to individuals' sexual orientation and gender identity. A lot of attention was given to this idea of transsexualism or transgender by this person here. Chastity Bono was born as a female to these two individuals here, Cher and Sonny Bono. These two were married, but they were also a musical group in the late 70s, 60s, and 70s, so well before your time. But they had a daughter, Chastity Bono, who was not comfortable with her gender identity at the time. And so Chastity Bono underwent a gender reassignment, took hormones, had breast removal and a reassignment surgery for his genitals, and is now going by the name Chaz Bono. So there's a book that he's written about it, and he's clearly much more comfortable with the male gender identity than he was as the female gender identity. But based on Chaz Bono's experiences, as well as what we've learned about the differences through neurobiology, it seems that gender-based neuroanatomical and functional differences may be flexible, right? So even though there are sex differences in males and females, there might be some flexibility to that gender structure. Even after developmental stages of sexual maturation, you can have administration of hormone and surgical alterations to alter gender-specific characteristics. So it seems that many of these gender-based variables that researchers have documented should be considered continuous rather than dichotomous, either male or female. So rather than seeing it as either male or female, we should look at gender as more of a continuum. So in other words, there are many shades of gray when it comes to sexual orientation and gender identity. So that's it for this lecture. This is the summary for this lecture. So animal models have been used to study the neuroscience of sexual behaviors and sexual encounters. The female sexual behavior seems to depend on the changing hormonal levels across the estrous cycle. Male sexual behavior can be exhausted except when new females are introduced, which was the Coolidge effect that we discussed. There seem to be sex differences within the brains of males and females, which may contribute to differences in behavior. There is evidence for a biological contribution to sexual orientation and transsexualism, but we need to evaluate it critically. It's unclear if this evidence that I presented to you is the cause or the result of sexual orientation or gender identity. So that's it for this lecture. Don't forget to submit your reflection.